but it's also a free radical scavenger and an antioxidant.
And an antiviral and an antibacterial. It's such an exciting product.
Correct. So, and it's almost too good to be true. You would say, no, how is it possible?
Sure. We have the privilege of welcoming Dr. Jacques Snyman here with us today. He's a doctor in immunopharmacology. He has extensive research in pharmacodynamics, clinical trial research and ethics.
Correct.
And you've lectured extensively worldwide on ethics.
Yeah.
Welcome.
Thank you.
We would like to ask you a couple of questions on the research that you've done on carbohydrate-derived fulvic acid. Tell me, what is carbohydrate-derived fulvic acid?
I think if we go back, we've been doing research on humic acids for decades. That was when I was still part of the Department of Pharmacology, University of Pretoria. And carbohydrate-derived fulvic acid came later as we moved away from the contaminated coal-derived ones to a more pure form where there's more consistency in production. And that's why we ended up with researching carbohydrate-derived fulvic acid as an immune modulator, anti-inflammatory, anti-infective product.
You're talking about the difference between coal-derived and sugar, plant-derived.
Correct, yeah.
Right.
So we must just remember humic acid's a natural substance, so it's a part of plant decay. So you'll find it in coal because it's basically a plant deposit.
So fossilized.
Correct. So you may get shilajit, which is a sort of a natural type, which comes from the Himalayas in the sense it leaks out of bogs, and then you have the coal-derived, which by also wet oxidation you form the new humic acids, which in fulvic acid is one of them. The problem with coal is it's also got anything else that goes with it, lead, mercury, and so forth, to such an extent that the Canadians now actually block the human use of any other form of fulvic acid because of the contamination.
That's why you prefer plant-based.
Plant-based means that you can control it. So you can make sure it's free from contaminants. Your input product is tested. It's manufactured in a GMP accredited facility, so you know it's free from any contaminant. Consistency in production is critical.
Can we pause there? What does a GMP facility look like?
A GMP facility needs to fulfill the criteria of your regulator. Now, our regulator is in line, the South African regulator, with overseas, like the European, Danish, FDA, and so forth. And to have a GMP facility, you need to follow the world standard in how you manufacture a pharmaceutical product. And that means that there's a particular process controls, and output that you need to keep all the way to maintain your GMP registration.
And that ensures standardization of the product and quality and, the temperature.
Everything needs to be well controlled and logged. So it's in a temperature controlled facility. It's in your products go through different phases of quarantine. Your output or your mother stock needs to be tested. Your output stock needs to be tested within specific standards, and it needs to have specific release properties. So your regulatory pharmacist would only release if it's within the certain specifications, which the criteria is very tight on.
So within this carbohydrate-derived fulvic acid, what are the benefits? What is inside there that has some kind of benefit to our health?
Your product is carbohydrate-derived fulvic acid. So it's the fulvic acid. So the carbohydrate just refers to the source and the consistency of the source and your particular fulvic acid. So fulvic acid.
So you're talking sugar as your source.
Sugar is your source, so you can control the type of sugar that you use, whether it's an organic sugar or a normal farmed sugar. It's usually better to move to environments that's well controlled, that you know what your source looks like.
So the plant is grown in an area free of heavy metals, and the water quality and the soil quality.
Correct.
You look at all of that before you source the plant.
Correct, and you need to remember that your processing into sugar also purifies your sugar. So
that's that incinerator that it's made in. It's nothing's going to survive that.
No, and you must remember the fulvic acid is made from sugar, and it's wet oxidation, but with the in a reactor where you then add oxygen, temperature with a heat pump. So you change all your carbons within your sugar into various specific acids. So it's an array of acid, malic acid, and so forth, which forms your fulvic acid. So fulvic acid is made up of lots of these, but it needs to be very consistently the same to get the same effect. Now that also differentiates from your, what I would say, mined or coal-derived type of fulvic acids, where the acid type changes dramatically, and therefore the effect changes. So we've done that, and we've sourced it on, you can source on the internet different fulvic acids, and you can then test the antimicrobial activity, for instance, to see is it different, and you can clearly see a difference between the different fulvic acids, so they're not all born the same. And that makes the difference in this particular product. It's tested, clinical studies on that, so we know the outcome, we know the clinical effect, and we know the safety of the product.
What is the mechanism of action of carbohydrate-derived fulvic acid within the body once consumed?
It's a very difficult question, because as soon as you imbibe fulvic acid, it gets absorbed and it goes through your gut lining. Now just remember, your gut is most probably your biggest immune organ in the body, and therefore you already get an effect there on the immune system, being an anti-inflammatory product, but it's also a free radical scavenger and an antioxidant.
And an antiviral and an antibacterial. It's such an exciting product.
Correct.
Isn't it?
So, and it's almost too good to be true. You would say, no, how is it possible?
Sure.
But what you can do is go Google those particular ingredients. You'll find that organic acids have anti-infective properties. We use them in the animal husbandry environment to treat salmonella, shigellosis. So it's not a new concept. The anti-inflammatory concept of this CHD-FA is very unique, and we've almost just stumbled on that. We use the anti-inflammatory effect to test whether the efficacy of our fulvic acid is optimal. And we've tested it in patients with eczema, in allergic rhinitis, in order to understand its mechanism of action. And you can clearly see, for instance, in allergic rhinitis, that you get a similar effect to your corticosteroids. So
Incredible.
Without the same side effects. So that if you look at the research, it spans from laboratory anti-infective, where we looked at antiviral, antifungal, antibacterial effects, and it's been tested in specific animal models in Rutgers University in the USA. An extensive, they got a USA government grant to study that. So very exciting research in wound healing there. Also in Birmingham and the UK MRC, significant research in infective conditions, but also as an anti-inflammatory. So because it is an anti-inflammatory, it allows, if you co-administer this with an antibiotic, normal antibiotic that we would use to treat a patient with, you would see that it has a synergistic effect, because your inflammation often protects against or protects your organism from being attacked by your antibiotic. It creates a sort of a protective environment. So if you reduces the inflammation around the organism, your antibiotic can actually reach it. We've done a very nice in vivo study in laboratory animals to demonstrate that effect.
But you're also looking at ingesting something that's good for the microbiome in the gut, which helps you while you're taking an antibiotic.
Correct. What it does, this is this notion, you need to weed the bad organisms and you need to feed the good organisms. And by creating, and now we're just talking acid, if you create a little bit of an acidic environment, you create a growth environment for the good organisms to grow. So you often see a good effect with probiotics combined with this. And because it's a scavenger, it's often scavenges the unwanted toxins that you also get in the gut. So not only anti-inflammatory, its scavenging effect is also helpful in the gut.
I think it's such a fantastic product. Just looking at safety profile though, I mean, you're talking about a sugar-derived product. Is it safe for diabetics?
Yeah, absolutely. You don't get any sugar at the end of the reaction, and I mean that's one of your GMP tests that you need to do, is that your mother substance is not present anymore, and it's all changed into a very stable, consistent fulvic acid pattern, very specific.
Right.
So there's no sugar left in it. Coincidentally, we've done a in vivo study at the MRC here in Cape Town, where we looked at diabetic monkeys, where they have a very specific model, and it works good. It brings down the or improves the control of the diabetes in those particular monkeys.
You're talking type 2 and 1?
Yeah, that was typical. It's a model that they have, which they then get rid of the pancreatic islet cells, and you then there's an insulin deficiency.
Sure.
So different degrees of insulin deficiency then either makes you type 1 or 2. So they've tested against sort of I would say, an intermediate model, so it would reflect more type 2 diabetes. So just remember, in the American Diabetic Association recommends organic acids as part of their diabetic control program.
Wow. But I mean, the wonderful thing about this product is all the different forms that you can use it. You can apply it topically, you can ingest it. There's sprays, there's gels, there's creams. What are the concerns? Are there any?
Not really. I think one must just remember this is not a miracle drug. It does what it does. It's anti-infective. It's an anti-inflammatory. And if you then translate that back into its efficacy, or where it's used, you will see that. So in eczema, it's an acid. So when you've got eczema, your pH of the skin rises, creates an environment for bacterial growth, so which then perpetuates the eczema.
So that's the secondary infection you can often get.
Correct. So now I put in on a slightly acidic product. You can't use it unbuffered, otherwise you'll burn. So it's slightly
that's the concern.
Yeah, slightly acidic, slightly buffered.
Right.
You put it on, and now
Gel
you
or cream?
Both. You can do both. We've formulated both. So what you will see is that you then change the pH, which then changes the environment. So you create an environment for healing an eczema, and then you bring in the anti-inflammatory component, which reduces the inflammation. So we've published on that. We did a good study on that, and a good, remarkable benefit for patients with eczema.
Can we talk about the taste? Because I've had products with carbohydrate-derived fulvic acid before, and it's a challenge to ingest it.
You can mask the taste a little bit, but that unfortunately it still comes through as not being too pleasant.
But how? I mean, if it takes away your symptoms, your cold and flu, and you can get up and go, take the taste and go with it.
Correct. The taste compared with the cold-derived is like chewing tar.
We don't want that.
You have that typical tar-like background, and it's also, you can almost, this is almost a translucent amber type of fluid. If you look at when it comes out of the reactor, whereas your other derived, especially your cold-derived, is almost blackish brown, and it's sludgy, so you can't see through it. So,
Good luck with your clinical studies, and please stay in touch.
We will. Thank you.
Thank you.